Yeah, good morning. Welcome to Compass on this Friday. Our speaker is Professor Claire Paris of the Department of Ocean Sciences. She has a bachelor's degree in biochemistry from the University of Bordeaux in France and a master's degree in biology and living resources from the University of Miami here, and a PhD in coastal oceanography from the State University of New York. And she has been a professor here since 2008. She started at the same time when I started. She is a biological oceanographer and has, according to her own biography, interdisciplinary expertise in marine ecology and Lagrangian dynamics. She's an expert for the dispersion and fate of larvae and marine pollutants. She has developed innovative Lagrangian approaches to track inertial particles. She has been involved in the modeling of deep sea oil spills, like the Deepwater Horizon accident several years ago. She has been the lead PI for oil spill modeling for the Sea Image Consortium within the GOMRI initiative. She has served as president of the early life history section of the American Fishery Society. She was in 2018 recipient of the AGU Rachel Carson Lecturer Award, and she was appointed to the Ocean Research Advisory Panel. And she is regarded as an authority on marine dispersal with all these things, and she has given at least 36 keynote speeches and invited talks. She has 138 peer-reviewed publications, 34 book chapters, and she has edited 2 books on the Deepwater Horizon legacy. And it's a real honor that she will give a presentation for us today about the coupled Lagrangian Earth system model framework for quantifying solar irradiance exposure on particles in the ocean.
Thank you, Roland, for this nice presentation. Introduction, I would say, because I'm giving the presentation, I guess. So thank you for being here on this Friday where everybody is more or less on vacation, but I think this is something that I presented at the School of Engineering a month ago, and it's a paper that is in review, and these are my co-authors. Robin Faitas is at the IFREMER in Lorient. Anna Carolina Vaz, she was postdoc, both of them were postdoc in my lab, and she's at NOAA. And then we collaborated with MBE, with Christina Kasparakis, who did a PhD here. And then Martin Rosenstiel, because we use this module to look at the effect of solar radiation in fish larvae. And everything is really under review right now. So why solar radiation is because it's a source of energy for life on Earth, but also an agent for photosynthesis, and it can be stressor at very high doses. And it varies with water optical characteristic, and this variability is expected to either increase or decrease, we don't know yet, with climate crisis. And the first step in predicting how such light-related process will affect the ocean is to quantify the exposure, especially by UV radiations, on living and non-living particles in the ocean. So here we see like a total irradiation. I don't know if I have to give a little bit of background on the solar radiation, but basically we have the radiation of the sun that go through the atmosphere. The atmosphere is a filter to it, but at some point it penetrates in the ocean, and when it reaches the top of the atmosphere, it's already it's already, you can divide it because the irradiance is measured as watts, units of watts per meter square, and because the ocean, the Earth is a sphere, you can divide it just by 4 the amount of energy or solar radiance that comes to the Earth. And so it's from 1,360 watts per meter square, it goes down to about 340 watts per meter square. But when you go to the ocean, it's really the short wave surface net radiation that matters, not all the wavelengths penetrate in the ocean. And this is a bit of a graph of the spectrum of solar radiation with wavelengths. So they're attenuated by water particles, water vapor, by or the atmosphere to begin with, and by the water as well. So I say that it varies spatially, and you can see that in the global distribution of incoming shortwave solar radiation. Average, this is average over the year from 1981 to 20. It also varies temporally. This is from a paper that we published back in 2021 looking at photooxidation of surface oil. And this is the amount of solar energy in the Gulf of Mexico at the Deepwater Horizon spill site. And you can see with the shaded area here that it decreases drastically when you have hurricane or, of course, cloud coverage. What we use is a NAVGEM, which is the Navy global distribution of shortwave radiation on the Earth, and this is really very useful because this is a model that is being validated with measurement from satellite, and it's 3 hourly, so this is what we're gonna use. For background also, the light does not penetrate very deep in the ocean, and again it depends on different wavelengths. What we've done with this modeling is looking really at attenuation coefficient also in the ocean, not only and by wavelengths. So I'll show you more about that. So what are the light-related processes? So there are many, even passive larvae, any particle that is constantly moving is moving in the horizontal, sometimes in the vertical, with turbulence or with their own movement. And this should be accounted for estimating the photosynthetic primary production, the PAR, also expose-related damage by UVA and UVB. So UVB does not penetrate as deep as UVA, but it does some damage as well. And the UV radiation, like UVA and UVB, also control the photosynthesis through regulation of various host interaction, and they can also inhibit the photosynthesis. So it's really important to understand how the particles accumulate this radiation, and so far there are no models that are looking at this aspect in a Lagrangian form and in 3D as well. There are protective mechanisms. Some of the larvae have a lot of pigment that can be stellate. Like you can see these fish larvae where those pigments can retract and expand with light. And then there is avoidance from the sea surface, where we've shown, and this is a paper in review with the Grossen lab, that there is buoyancy changes even in the embryo of fishes when they're exposed to UV radiation. So most marine species move, at least vertically, in the water column, while biotic particle vertical velocity are also dictated by their physicochemical characteristics. So here we're looking at the abiotic particles, like hydrocarbon. Oil can be photooxidized, microplastic can be photodegraded. And so the role of the UV radiation spreads beyond biology. It's the exposure increase the fragmentation rate and photodegradation of plastic and microplastic. And like I say, it's key processes in the photooxidation of the transformation of oil slick that become even more toxic. So over the next decade, net primary production of fisheries is predicted to decrease, while plastic concentration and the frequency of oil spill will increase. And these changes will coincide with the increase of UVR, perhaps. But there was no tool that was developed for that, and here we present this modeling framework that estimates the exposure from an individual-based perspective. So how do we get the data? The data from our satellite data provide accurate snapshot of the irradiance distribution at very high resolution. Several studies examine the penetration of light in the water column, in situ measurement of UVR or PAR at fixed station, predict the level of photoinhibition in primary production, and also the exposure of tropical coral reef and fish larvae. So we have other satellite data that generate the global maps of average UVR irradiance and potential DNA exposure as well. So this is really important now that we're looking at eDNA in the ocean for biodiversity, and there's actually a student that is visiting my lab is coming today from Ifremer that is looking at backtracking eDNA in the ocean. So the validity of the predictor exposure is compromised by the steadiness over time. So right now what they do in the model, they have like a value, and they use this value almost globally. This is starting the methods section now, where this is our modeling framework, and we're closing the gap by doing this four-dimensional biophysical model. And we use the Navy Global Environmental Model, NAVGEM, and the Mercator Global One Force model. So the model that we developed was initially at very low resolution because we wanted to see globally how the irradiance changed among oceans. So, and we use the UVA in the upper ocean from the in situ BGC floats to estimate the UVA penetration in the water column, and we developed this Irradiance Module to calculate the irradiance at depth. So we have the irradiance, this exponential decline basically. What is important here is that all these values are given by the Z value, which is the depth by the particle movement. And the irradiance is given by the attenuation coefficient calculation. And this is really the bottleneck because there's not too many attenuation coefficient calculation done. And right now I've heard from last couple of weeks that there is a satellite that will measure, will be able to measure attenuation coefficient in the water, which based on the clarity of the water based on the phytoplankton production and particles scattering the light in the water. So this is the module that we developed, the red one here. I'm going to talk a little bit more about it. Okay, so if we look at the different part of the model, you see that there is a module that is using the ocean circulation model, and with this ocean circulation model that you see in the background, we can either calculate the concentration of the particles or the individual trajectories of particles. So this is very flexible depending on what we want to do. This is open source model in GitHub. The hydrodynamic model, like I say, is the Mercator Global 26 kilometer horizontal resolution. It has data simulation that has sea surface temperature data, drifting buoy data, and it has a lot of vertical levels above the 200 meters, and we're really looking at irradiance in the upper ocean, so 200 meters is enough. For the irradiance module, the input data is then the surface irradiance and the coefficient of attenuation, like I said before. So the irradiance module is flexible regarding the source and the unit of the surface irradiance data. The data should be organized in NetCDF files with time, longitude, and latitude, and the light penetration through the water column is determined by the attenuation coefficient KD from the Beer-Lambert's law. The coefficient differ in water mass and region and can be spatially temporal explicit, following the same grid resolution of the irradiance data. And so the coefficient is defined by the user, and it allows to use either theoretical model or empirical observation, such as UV light penetration from the BGC float. So here in this map, what you see is the different coefficient of attenuation that we use and the variability. There is not too much variability in the season, so we use an average value. We use an average value. The largest difference was the coefficient of attenuation between oceans, where you see that in the Nordic seas, of course, the attenuation is much larger than it would be in the Pacific Ocean. So this location will be the release point from our Lagrangian particles, because these are the location where the float data were taken for the accurate measure of the coefficient of attenuation. So for solar irradiance data, we use then the Navy Global Environmental Model, the NAVGEM, with shortwave surface radiation, and the 3 hourly. And these are tables, I'm sorry, it's not very clear. But for the attenuation coefficient computation, we use both SPAR and UVR from the vertical profile, and we did the mean variability attenuation for the Nordic Sea, the Sargassum Sea, the Eastern Equatorial Atlantic, the Equatorial Pacific, and the Southern Indian Ocean. And for each region, it was computed as the slope of the linear regression of the radiation measurement of the specific wavelengths. For example, if it's UVA 380, so with depth and pressure throughout each vertical profile for all months for which the data were available, and then we took the average of this value. Like I said, seasonal variability observed at it was relatively stable for PAR and UVA in this region, so we took the mean annual values. Okay. So these are the release location of the particle. So what are the simulation scenarios that we use? Like I said, we want to mimic biological particles and abiotic particles. So we did passive particle just as a standard scenario where the particles were just neutral buoyant and released at different depths. Of course, even though they're released at different depths, they're moved by the current, so they are not staying at those depths. The biological particles, we mimic kind of like the zooplankton with diel vertical migration. And for the abiotic particles, they were neutrally buoyant particles that persist in the water for a long time period. So for the biotic particles, the release and the tracking was only 7 days, but for the microplastic particle or the debris particles, they were tracked for a year. Okay, some results. So here what we are showing is the mean and standard deviation of the cumulative dose of UVA over the 7 days of simulation for each of the region. And we consider the different particle behavior. So DVM is in yellow, and the different depths are in blue in green from 1 meter, and dark blue for the deepest one. So even though like the UV dose are similar between the Eastern and the Atlantic region and the Pacific, while the irradiance is higher in the Eastern Atlantic, the UV dose are much lower in the Western Atlantic compared to the Pacific. So this pattern results from the regional and seasonal difference in UV irradiance reaching the ocean surface. So we see a very large difference into the mean irradiance from those particles, and also regional difference from the different oceans. On the graph on the side is the mean and standard deviation for all the seasons combined per region, ordered latitudinally. So you can see the latitudinal gradient of the surface of UVA with a gray area. So it's a little bit what we expected. So what we want to do now is looking at what is happening at a smaller time scale. Interesting enough, there is a lot of variability, mostly in the Atlantic and the Pacific Ocean. So what we did now is at the finer temporal scale, we look at the daily duration of exposure in the subpolar region. On the top, so you have the North Sea, West Atlantic, East Atlantic, Pacific, and South Indian Ocean. On the x-axis is the local time in day, and everything is averaged for these 4 months, January, April, and July, and October. These are the months that we release those particles. So it's expected that, of course, in the North Sea, there's not much light at all, so there is not much variability. What is interesting is that the DVM, so the diavertical migration, what it does really is reduce the variability in exposure, or the shaded area are the variability. So even though the DVM looks like it's 15 meters, the variability is very much reduced. So we can say that biotic particles are adapted to solar irradiance by trying to reduce this variability as compared to staying at the same depth. Of course, there is a lot more trade-offs from feeding and other trade-offs, but in terms of solar irradiance, it's a very interesting result. So the vertical difference in exposure are notably different between regions. There is homogeneity, throughout the water column in the Pacific and the Southern Ocean. And what we found is that the exposure of UV radiation is primarily determined by the 3D pathway rather than by the irradiance reaching the ocean surface. So if you overlook this motion of particles, you cannot accurately predict how much irradiance they receive. So let's look at now the pollutant particle. We steer away from microplastic, and it's more like pollutant in general, because if you wanted to model microplastic, there is a lot of faith involved into it, and we just look at the solar irradiance. And so these represent persistent pollutant release on the 24-degree parallel, passing through each 0.5 degree cell for a whole year. So they will release not to the west or to the east, but along this parallel. And then we can see on the top, on this graph, the top graph, this is the average. Yeah, this is the mean solar irradiance for all the trajectories. And the bottom graph is we selected a few trajectories to see what is happening, and for the paper actually we're working on doing the average over the months to see what we suspected that we see here. What is interesting is the biotic particle, drifted throughout a large portion of the North Atlantic, from Cape Verde to Belize and from Venezuela to the south of Greenland. So that even though they were released in one parallel, they drift quite away from that release site. The highest mean cumulative UV exposure occur in the Eastern Atlantic between the 25 north and 38 north. And to a lower extent in the Atlantic region until the US coast. So the variability in exposure of this zone cannot explain the higher mean exposure, as the distribution of the standard deviation is more homogeneous than the mean. I don't have the standard deviation here in this figure, but the only way to understand what is happening is by looking at the vertical position of these particles. And here what we see is in the particles that there's only 3 trajectories. So you can see one that is released near the surface. It has about the same at the end, cumulative, only it doesn't have more cumulative irradiance than the one that are released deeper. So it is counterintuitive, and we're trying to understand why that is. Besides the areas of higher expect cumulative UV exposure, the particles drifting towards the equator receive among the lowest UV exposure. The pattern results from the vertical distribution of the particle because of the currents that are taking this particle further down compared to moving north and going further near the surface, so in the upper ocean. So this is why it's important to include the Lagrangian scheme into the accumulation of solar irradiance in the particles. So here, actually, in this one you see the variability. So the variability is very large. So these are our results so far. We are working on fishery application, where we model the embryo response to UV in the lab, and we're putting that into a Lagrangian model. And what we see is, due to the specific gravity, the embryos are only reaching the surface in the winter, and that is when the UV exposure is at its lowest. So these embryos are adapted to get to levels where the UV is not as high, which has implication of what happens during climate change, because if there are any change in the environment, then those larvae may have like a mismatch with the food if they are moving deeper than they usually do. So that has some implication on that. And this is really the first time that we show that those embryos are not at the surface, which for me was really intuitive, because they don't have pigment and they are not adapted to UV. So if they were to be at the surface, they would not survive. And here, because all the models in fisheries are showing all the eggs at the surface and tracking the egg at the surface, but not really at the surface. And it does change where they go. The other application is the oil spill application. This was during the Deepwater Horizon spill with the CMH consortium. And what we did here was looking at this module coupled to the oil spill to look at photooxidation. So after an oil spill, oil droplet in the ocean surface can be transformed by weathering processes. Let's see if I can see it here. And it's called the POCs. These are photooxidation components. And here we see the photooxidation with the red particles and the rest of the oil spill. So the photooxidation components are really close to where the oil will spill, and that reduced the degradation of crude oil by exposure to light and oxygen into a new product over time. So tar, for example, is a byproduct of this weathering process and can remain in coastal areas for decades. But despite the consequences of the weathering pathway, photooxidation was not taken into account in the oil spill, like the Deepwater Horizon model, and in the budget calculation either. So we developed this first oil spill model algorithm that tracks the dose of solar radiation on the oil droplets. And we demonstrate that under realistic environmental conditions, the ocean after the Deepwater Horizon spill photooxidizes into these components. This is the first model that supports new paradigm of photooxidation that emerged from laboratory research. And what is more is that the photooxidized oil is known to reduce the effectiveness of the dispersants that are deployed on the oil slicks. So that means that if you deploy a lot of dispersants in areas where it's already photooxidized, then you're not really, I don't know why it's not moving here. So we found that the weathering of oil droplet by solar light occur within hours to days, and that roughly 75% of the photooxidation during the deep-water Rosenstiel spill occur in the same areas where chemical dispersant was sprayed from the aircraft. So here you see a picture of the red lines where the dispersant was sprayed, and the circles are basically these 75 threshold for high solar radiation and low solar radiation, but in all the case, the dispersant were used in places where it was not photooxidized yet, but beyond those circles it is photooxidized. So that allows to have a more precise idea of where to put the dispersant if you have to spray dispersant near the surface. Okay, so moving forward, there are many biological, chemical, and physical applications for looking at irradiance at the surface of the ocean and in 3 dimensions on moving particles. We have advanced in computational capability, and we know that solar energy is vital to not only photosynthetic organisms but triggers several other light-dependent processes. Biogeochemical application can range from, like we talked about, the photooxidation, but also the phytoplankton in the austral ocean, looking at what happens to the blooms, photodegradation, and those in this process are currently omitted in oil dispersal and toxicity model and in photosynthesis model. So I think that it has a lot of implication for future research. So if I want to summarize, I think I went a little fast, but it's okay. So we can have more questions. Solar energy is vital for many, several light-dependent processes in the ocean, of course, even though the light doesn't penetrate that much in the ocean is not transparent to light. There is a lot of processes that are critical for the upper ocean. We saw that over long exposure of UV dose, UV radiation is a stressor for fish population through lethal effect on the early life stages. So now we have not only to worry about pollutant in the ocean, but what is a combined effect of ocean pollution and change in solar radiation. There is a decrease in precipitation that will also increase the UVR locally from IPCC report. So the development of fully coupled biophysical atmospheric model will become important to understand species and particles response in changing conditions. There are other environmental conditions changing, like salinity and temperature, that often take into account solar irradiance, and UV exposure have been so far not considered. So we do fill in this gap, and we're looking forward to collaborate with other labs to test our hypothesis of the importance of these light-induced processes in the ocean. And with that, this is a paper here that we have in review. with Robin Faita's leading, and we thank Mathieu and his gang for the NAVGEM data, and this was possible with Recover2 and CIMAGE grant from the Research Gulf of Mexico Research Initiative. Thank you.
Yeah, thank you very much. Questions from the audience here? Igor, I will come with my microphone.
Wonderful talk. Thank you. I have a question about the part where you talked about the particles. Have you only considered neutrally moving particles, or how about positively moving particles that accumulate on the surface, for example?
Yeah, for the abiotic particle, we just consider neutral. But we did release at 1 meter. We did several releases at different depths. So, but we didn't look at the changes of depth with particle, except for the DVM, the diavertical migration, where they really mimic the velocity of descent and ascent in the water column of copepods. But yeah, you can do a lot of different release and see how it changes, but altogether what we found is that if you release a particle just at 1 meter at the surface and compare it to other particles that are moving vertically, it changes, of course, a lot the irradiance. But even for the particles that are at 1 meter and that are moving, makes a huge difference by I think it's an order of magnitude in terms of accurately capturing what is a cumulative irradiance on this particle when you use the Lagrangian scheme versus like a static scheme and looking at what is happening in the ocean.
Do you have a sense of how solar radiation is changing over the surface of the Gulf of Mexico over, say, the last 5 decades or so? It's been warming a lot.
There is a graph that I didn't show here that shows this seasonality. There's data that we're taking from back in the 80s to now. But I haven't looked at the trends. But it's possible to look at the trends to have an idea of that. But I haven't done that. But there are data that have been published looking at this value measured from satellite from back in the 80s to today.
More questions?
What we don't have as measurement are the coefficient of attenuation, which again, that can change. It's not only what comes from the surface, but what is happening in the ocean. Because if you have less primary production, then this coefficient of attenuation are going to change, and then the solar radiation are going to penetrate further down. So we don't know what is the synergy there, because we just put this out, and we use it for a couple of applications, but we haven't really fully looked at those larger questions. Yes.
Shane?
Okay. So I think you get the velocity field from a global model, right?
Yes.
A dynamic model. But it has a limited vertical resolution, right? Close to the surface. So you must be doing some interpolation of the velocity.
Yes.
close to the surface. So I'm just wondering what type of calculation do you use and how does that affect the result?
That's a good question. We have like a tricubic interpolation, so we're looking basically at 64 points around the velocity field. So even at this 24 kilometer resolution is still accurate in terms of the tracking the integration scheme for the velocity.
What about in the vertical?
But in the vertical, we only look at like the 200 meters. We're not looking at, and that's where you have most of the layers in this model. So, but yeah, it's we haven't tested in higher vertical. I think it's a good point.
So what are the
So
first,
if you were a reviewer, you would say.
so but what are the first layers in your global model? Like 1 meter, 5 meter, what is it?
Yeah. Wait. This is here. 0, 5, 10, 15, 20, 25, 30, 40, 50, blah, blah, blah.
So your particles, they must be moving very little in vertical, right, at every time step. So they don't jump from 0 to 5 to 10 and back up, right? They go at intermediate depth. Is that correct?
They, we take very small steps because we only track them for 7 days in these cases, except for the one we track them for like 100 days. But we use vertical diffusivity according to Kubo, so like bigger the layer, like higher the vertical velocity is. So we kind of like take care of that with the vertical turbulence. Thank you.
This might be a better question for your co-authors, so sorry about that, but I'm just wondering if you could briefly tell us with the embryo UV avoidance, how you know it's due to the UV rather than like predator avoidance.
I can answer that actually because the tests were done in my lab looking at the effect like in a tank vertical column that is 7 feet tall, and we have a ceiling with light, with UV light, and we measure the UV light and we put embryos there and expose them to light. That was Christina's work. Yeah, so we knew that they would change the
But would that also
buoyancy.
be an adaptive response to the presence of predators in high light conditions? Or an
Say again?
evolutionary- Could that also be an evolutionary response to the likelihood of predators under high light conditions?
Yes. It's true. And then those embryos were put in the a column, like a buoyancy column, to look at the change of buoyancy with the UV light. So we know it's directly triggered by the UV light because when we measure their buoyancy, they had change, and it's like, yeah, you're right. That I can answer what is the mechanism inside the embryo, where they release some ion or something to be less buoyant or more buoyant. But it was triggered by the exposure to light, for sure. And yes, it may be like a adaptation for avoiding predation because the diaphragmatic migration happens with moving down during the day. But even though during the day, when we put these values into the model, we saw that the embryos, even in the day, where they're moving up and down the water column depending on the irradiance of they were exposed.
Okay, one more from Beth.
Following up on that, do the embryos remain sensitive to UV until they grow up into an adult fish, or is there some point where that sensitivity just goes away as they become older and less vulnerable to light?
So the embryos are until hatch, so it's 48 hours. And then when they hatch, the preflection larvae are pretty clear, and the pigment develops through ontogeny. So, so nobody has done this for preflection larvae to see how they would avoid light, because interestingly, fish larvae are attracted to light. We did some experiment with that too. But I always wanted to do this because, like I show in some other slides here, this is a fish larvae with a lot of pigment, and this is a sand larva during the night. So during the night, and the pigment are the melanophores, and they are called stellates because they look like stars. And so the pigment, the melanophore inside the pigment pocket is concentrated during the night, and you even think it's like a different larvae. So I'm pretty sure that, you know, like neustonic larvae, like larvae from Exocetidae, like flying fish and stuff like that, they're very pigmented. All neustonic larvae have a lot of pigment. And if you find a larvae that has very little pigment, like mesopelagic fishes larvae, or even scarab larvae, like parrotfish larvae, they have no pigment whatsoever. They are in the deep water column, like at 80, 100 meters, sometimes even 300 meters. So I'm sure nothing has been done in that sense, but I think it would be a good topic of research for sure.
Okay, were there any questions from the Zoom audience? I don't think so, but if you have a question, turn your camera on and speak. But nothing happens. Then thank you again, Claire. Thank you all for coming. Come again on Wednesday when Junfei Xia talks about his PhD work in SLAB 103, MPO, 1 hour student seminar. And next Friday we have Abhishek Zavita, new postdoc in APM, talking about something that he wanted to talk about this Wednesday, but we have to postpone it because of the hurricane. Have a nice day.